Hi everyone, my name is John. I'm a business development manager here for Celemics. For those of you who are unaware of who we are, Celemics is a library preparation and target capture panel manufacturer who specializes in human oncology and inherited disease related panels. So the agenda for this presentation is for me to give you a brief introduction as to who we are, what we do, and then what we have to offer. And then at the end, I wanted to give you a specific case example as to the capture performance of one of our panels compared to the other panels seen in the NGS market. So what is Celemics' role within the NGS industry? We're involved in the manufacture of reagents and bioinformatics services, and with these products and services, our main targets are hospitals, clinical labs, CROs, in which they would use our panels as IVD tests. Aside from them, we also target research and academic labs as well. When it comes to our target enrichment kits, we mainly focus on the human oncology and inherited disease sector. However, with our probe design and synthesis technology, we have also made panels that apply to the non-human sector as well. So now the big question is, how does Celemics differentiate themselves with the big market leaders? Well, I wrote down some points regarding Celemics' capture technology as well as some advantage points that we hold. So starting from the top, our target enrichment kits are hybridization-based capture, and with that comes high coverage and uniformity. And with the performance of our panels, researchers can discover all variant types such as copy number variant, insertion deletions, fusions, MSI, TMB. And then we also perform what we call a pre-performance validation for every customized panel. And what a pre-performance validation is for every customized panel that a customer orders, we'll test the probes against 3 HapMap samples to ensure the capture performance for every target region is optimal. And with this data, we'll send it back to the customer for them to either give us a green light or a red light. Green light meaning, yes, you could proceed with the manufacture of this panel and send it over to us, or a red light in which, in those rare cases, they will request us to do a probe rebalancing service in which we will test the probes again, rebalance the probes, and then send that data back to the customer for them to reassess once more. So a lot of customers are satisfied and find value in this type of service that we provide free of charge. And then lastly, as a manufacturer of all of our reagents, we have a wider price flexibility compared to some of the market leaders out there. And as a company that only has around 60 employees, we have over 20 scientists within our company, which means we can provide the best technical troubleshooting support in the market. So this slide is just to indicate the performance of the Celemics panels compared to our competitors. Utilizing our proprietary probe design technology, our capture performance consistently enables superior uniformity of target capture compared to our competitor counterparts. Another example, even when it comes to hard to capture regions such as GC-rich regions, as shown below, or AT-rich regions, homologous regions, our probe design optimization enables superior capture in these types of instances. As mentioned prior, our main expertise is in the design and manufacture of human oncology and inherited disease type applications. And although we have a wide variety of different off-the-shelf type panels, we have the capability of designing and manufacturing customized panels as well, which is actually our main bread and butter as a company. So as you can see from the left-hand side, the target enrichment portion, we have a wide variety of different types of off-the-shelf panels, smaller cancer panels with 10 genes or less, larger comprehensive cancer panels with over 700 genes. We have a whole exome sequencing panel with a size of 37.1 megabases in size, ctDNA panels with supported UMI systems, so a wide variety of different types of panels. However, for this presentation, I wanted to emphasize on the clinical exome research panel that we have. So we have 2 clinical exome research panels. Our standard panel has a panel size of 13.8 megabases, while our expanded panel has a size of 19.6. So as our clinical research exome panel is focused on sequencing only the most clinically important genes, it's an excellent tool for a comprehensive genome profiling for a wide variety of different genetic diseases. So as a capture, performance is extremely vital, especially when it comes to disease-based research. We have optimized our panels to ensure the best capture performance. And with better capture performance, obviously comes improved disease identification and the study for disease pathways and mechanisms. So in the next couple of slides, I'll show you some instances where we have compared our clinical research exome panel compared to similar competitor panels. So this particular case is showing the capture at the SUZ12 gene. And as you can see, when it comes to the Celemics panel, we have captured all exons. However, with our competitors, you can see there are 6 SUZ12 exons that are not captured at all using our competitor panels. Within the same panel, there were 2 Casper2 exons that were not captured within company B's panel, and then 9 exons that were not captured using company A's panel. A little bit more of the same story with the genes below, where many of the exons for these disease-related genes were not captured at all using the competitor panels. However, using the Celemics panel, we successfully captured all these disease-related targets. So when it comes to clinical research, it's extremely important to correctly sequence all target regions with high coverage and uniformity, as many of these target regions contain variants that are linked to genetic disorders. So the last 4 slides were just to indicate that with our clinical research exome panel, we have only the most clinically relevant genes. And within those clinically relevant genes, we have superior capture performance compared to our competitor counterparts. So that's what I wanted to indicate in some of the case examples that I showed. So lastly, if you have any questions about our technology, our capture panel portfolio, or if you have any specific NGS projects that you want us to consult you in, please don't hesitate to contact us. Our US office is located on the West Coast, specifically in Corona, California. And then we also have branch offices in Bangalore, India. And lastly, our headquarters is in Seoul, South Korea. So thanks for listening in. I hope you have a better understanding as to Celemics as a company. And once again, please feel free to contact us with any questions. Thank you.